Good morning, everyone. So I will start the first talk for today, multi-omics. So what is multi-omics and why do we care? So basically, this is the central dogma of molecular biology. In our cells, and the DNA gets transcribed to RNA, and they're translated into proteins. And this is something we all learned during our high school. The information has been converted at different omics level. And here is a detailed workflow of the process of transcription, translation, and corresponding techniques. From here, you can see the information is just flowing in between different omics level. But can we really infer the information, for example, the protein expression just based on the information from the DNA or the RNA level? So these days, probably you have seen this figure from time to time. So in this cross-omics comparison, you can see that the correlation of different omics level are not really so good. Like, for example, between the transcriptomics and proteomics, the R-square is just 0.36. And even if you use ribosome profiling techniques, which is showed in this figure, you basically just extract these parts of mRNA that actually bind to the ribosome and sequence them. You think probably they all get translated and converted to protein probably, but here you can see the R-square is just 0.57, so it shows that we cannot really get the functional information just based on the transcriptomics or even translatomics. And here there is another example to show you the inconsistency between the omics. So here chaperone is a very important protein for the protein folding. So after the protein chain gets translated, they will need to get further folded into the functional protein. So in this study, they knock out the chaperone protein. So you can see here, there are the expression level of several proteins are significantly lower, and the correlation is not so nice. So from here, you might think that proteomics is very good. It's much better than other omics, but for proteomics, we still need the support from other omics, and they're better together. So here I will show you a slide I steal from Perseus slides. This is a procedure for the database search. And here you can see that we need a reference database for the database search. So basically in this workflow, we already showed you several days before. We use a protein database from the UniProt database and do in silico digestion and revert it for the FDR control. And at the end, we got the result for the database search. But if you think about it, what if I cannot get a database reference protein from the UniProt? For example, what if your samples, you don't know the composition of your samples, like microbial, or you are studying some organisms that are not really a model organism that has been recorded in public database, or like, for example, for most of the microorganisms, you can actually find the reference proteome from the public database like UniProt, but actually this reference proteome are not really like experimentally verified. They're just predicted from their genome sequencing. So In this case, we will need the support from the NGS to create your own database, maybe if you want to get better identification. And another situation is sometimes you get samples with genetic variation. It's not just something like cancer or tumor, but like its genetic variation is commonly exists everywhere. Like, for example, my genome is different from yours, and of course, ours are different from the reference genome from the public database. So sometimes if you are particularly interested in some genomic variations, you might need the help of NGS. So sometimes at the DNA level, there is some mutation or substitution. And here is an illustration of this mutation. And some nucleotides mutation won't really change the corresponding amino acids because of the codon degeneracy. That's what accounts for the existence of synonymous mutations. So people might ignore it since it won't really change the amino acids, and so it will not change the protein sequence as well. However, some mutations might introduce a stop codon instead of amino acids, or it will turn into another amino acid. So these are non-synonymous substitution. So they matters because they might change the protein function and further affect the biological process in the end. So for standard shotgun proteomics data analysis pipelines, usually we only identified peptides that are encoded in the reference genome. So in some situation, like for example, for some researchers studying cancers, maybe for them it's desirable to identify peptides resulting from the non-synonymous variations as well. So if you are expert in NGS, congratulations, it's not a problem for you at all. You can assemble your own customized protein database for the database search by yourself. But for researchers that have less experience in NGS, Pavel and Max, they built a module. Pavel was a PhD student and postdoc from our group, and Max was a master's student. And they built a module in MaxQuant that takes both DNA and RNA-based NGS data as input to create a customized database in a highly standard workflow. So you can actually like comparable between your different studies. And this allows for the identification of variant peptides that are otherwise missed in traditional workflow. So this is the workflow of the new module. So the first step is variant extraction. So first we read some NGS data. This is a screenshot of the module. So to use this module, you can open MaxQuant and navigate to the tool tabs to select variation extraction from the drop-down menu. And also here you can see 6-frame translation, but I'm not going to introduce it, just to let you know if some of you want to use it here. And once you click the variant extraction, there's a new pop-up window, and under the raw files, you can load a folder containing the BAM files. So for us, at the moment, we don't support .fastq file. We support the BAM files from which the variants can be called. So the default calling parameters are optimal for the most common applications. So in most cases, you can just use the default setting. And under the global parameters, you need to specify the corresponding genomic reference sequence in the FASTA format here under the sequence file, and genome annotation in GTF format under the annotation. And also you can provide the desired directories where the temporary files and also the output files will be generated. And optionally, a VCF file can be supplied, which is this variant files to annotate the detected variants. So after you run this module, this is something that happened before the database search. It will create a folder in the specified directory, and it will contain 5 files you can see from here. And the most important one is a proteins.fa file. It's a FASTA file that gives the IDs of the proteins and their amino acid sequence. And if their particular proteins contains variants, the ID of these variants. And for the second and the third file, basically they will not really be used in your database search. So they contain some information that represents the identified variants. You can check them for further information. And there are also 2 XML files. They contain the information about the parameters which have been used for extraction, and cumulative statistics about the amount of initial and filtered fragments in BAM file. Then after we extract all these variants, we get a new database, a customized database. Then we can move to the next step. So next, now we have a protein database with extracted genomic variation. We can do the database search with this database, and you will need to modify some parameters for the sequence database to activate this. So this is the interface for select the database. Basically, you go to the global parameters, click sequence, and you can do what we showed you. In previous days, you can load a database. So in this case, you load this proteins.fa file that has been created from the module. And after you do this, don't just start the database search. First of all, you need to extract the variation rules. So you need to click this variation rule. So basically, we already create a regular expression to extract all the information we created for the variation in our protein database. So you can click it and just click OK if this database is created by this module. Then after you click this, if you want to see if they are extracted properly, you can click the test. Then you will see there is an extra column for this variant information. And before you start the database search, you also have to change this option. So by default, it's none. And to activate this, you need to click none and select read from the FASTA file. So after you specify the variation rule and change the variation mode to read from the FASTA file, you can start the database search. And yeah, and now you will, so in your output files in this peptide.txt file, you will get 2 new columns that contains information about the genomic variation. One column is called mutated. It's basically only contains information about yes, no, or mixed about the genomic variation. And there's another column to show you where exactly or which is the genomic variation exactly. So you get extra information in this peptide.txt file. So in this study, I show you from the publication from Powell and Max. They test this module with a data set that was generated by another study. They generated deep proteomes of HeLa cell line on its multi-enzyme. So trypsin, chymotrypsin, lysine, and the combination of them. So with this workflow, we were able to quantify the unique variants identified by a single or combination of different proteases. And this figure showed non-synonymous variants that's seen in different combination of proteases. So with this figure, we can see that with trypsin, we are able to identify more unique peptides, but you can also notice that for better coverage of proteome, it's better to use more than one protease. So here we show that with our workflow, we can identify genetic variants. And how about comparing with other omics level? They also did RNA-seq. So this figure shows the frequency of substitutions observed in the data at proteomics and transcriptomics level. And the color-coded proteomic detection rate show the proportion of the identification on the proteomic versus transcriptomic level, and the median proteomics detection rate of the variance is about 30%. And you can see that the amino acid changes are not uniformly distributed among all possible changes. Like you can see different colors, different size, and this is probably due to the different frequencies of amino acid and genetic code related restrictions, but also possibly it's because of the physiochemical nature of the amino acids. So if you really want to know what's happening on the biological process at protein level, it might be a good idea to just not be the best idea to just infer this information from just genomic or transcriptomic level. And now here is a reminder for the presentation from Pavel this afternoon. He's going to introduce his work about the global detection of human variants and isoforms by deep proteome sequencing, which he also used this module in his research, and he also used multi-protease to digest the proteins. And at the end, they also profiled proteins and transcripts variants in a website where users can really explore the protein isoforms by themselves. So please don't miss it. It will be a great talk. So basically, this is a workflow for gene expression analysis. Excuse me. And you have your reads from your sequencing machine, and you align them to the genome. You can see that. Then you can annotate them, and by the end, check the gene expression table. And at the moment, we cannot do this part from our software, but we managed to add support to start with a .bam file and to create a gene expression table with Perseus. And here I can show you how we can do this. So when you go to Perseus, there is an icon here. It's for NGS data upload. And if you click here, there is a pop-up window. And here you can book your BAM file here. And then you can basically get a table where for every gene, you get a number that tells you how highly it is expressed. And so for here, just to remind you that you can only load the NGS file in BAM format. Thank you. And then you can, here there is a parameter, strength or specificity. And for this option, it's basically you get this information from those people that do the genome sequencing for you. It shows the direction for the sequencing. So after you specify the parameter. You click OK, you get the gene expression table. And under this menu, if you click here, the gene expression, there is actually another function called isoform expression. And basically from here, you can take the transcriptomics file, the BAM file, and also the peptide.txt file that you created, or you get from the database search from MaxQuant. And for Perseus, it can map between these two files and create the results for the isoform expression. So what else can we just go beyond the central dogma? So the first thing many people might think about is metabolomics. Metabolomics is quite challenging. It's not part of the central dogma because for all these metabolites, you don't really have sequence, so you cannot really map with protein or RNA or DNA. But for us, we have created a Perseus plugin Metis, which you can combine the result from other omics level with from the central dogma with the metabolomics data, and we can map them together at the pathway level, so we can do the analysis. And this work was done by Hamid and Danila. They are not going to introduce it due to the time limit. So you can, for more details, you can check this publication. And this Metis plugin is already loaded into the newest version of Perseus. And what else can we do? So now microbiome is a very hot topic. So it's almost correlated with everything, and not only for health, but also for environment. And here we're trying to study the Metaproteome, and Metaproteome is a proteome of complex biological microbial communities. It's really challenging. So first of all, for microbiota sample, you don't really know the composition of them. So it's very hard to construct a proper protein database because you don't know which kind of species or genus is inside. And the second tricky part is a huge search space because it's so complex to identify all these proteins. You might need to put a lot of proteins inside the database, so the search space will become huge. And also because of the huge search space, you got some FDR control problem because you have a huge database. Your reverse database is also very big. Then you get a very high probability to get this false positive match. So here, our group, and especially me, I'm working in integrate a new module just for metaproteomics analysis in MaxQuant. And I think next year we should be able to produce a tutorial or a presentation for this module. And if you are interested in this, you can also discuss with me later. So what else? We can also think about other omics data and probably also some imaging data like fMRI scans or something else. And there are a lot of possibilities. And our dream is to integrate the analysis of everything into MaxQuant and Perseus. And now we are working hard to make this dream come true. And